Right. Yeah, thank you very much. I'm very pleased to be here, so thanks for the invitation. My postdoc, Nora Hangel, and I spent several months analyzing interviews that she had conducted with a number of scientists. In these interviews, scientists didn't have many positive things to say about failure. They tended to associate failed project with personal failures, and especially early career scientists worried about future unemployment. When you Google failure, you will find a lot of motivational wallpapers that tell us that failure is actually something that we should embrace and it's a good thing. So that is more in line with the conference theme. And what I want to do in my contribution is I want to suggest that studies of actual scientific episodes can do an even better job in reassuring the scientists and maybe preparing them for life, as was mentioned earlier, than these motivational wallpapers. So they can tell us more about what failures actually are. Now, I would like to illustrate that with just one example from the early history of bacteriology. So in the mid-19th century, that is prior to the big medical investigations of bacteriology. So I want to look at botanist Ernst Hallier's theory of pleomorphism. It was called retarding advancement. It was called a pure fantasy, fortunately refuted. It was called an absurdity, and even recently somebody talked about Hallier's murky implications. So we can see in the eyes of the commentators, it seems to be a rather big failure. Now, when we look at the actual historical situation, so these questions arise. What kind of failure was this? What, if anything, did 19th century bacteriologists learn from it? And what, if anything, can we learn from it? So, just a few words on bacteriology prior to the 1880s. So that was a time when bacteriology was part of botany and zoology rather than medicine. And researchers, predominantly microscopists, debated almost everything about bacteria, what they were, whether they were plants or animals, and how they could be classified, how they developed, what they ate, how they slept, what food they consumed, what effects they caused, and so on. So everything was kind of in doubt. So the idea of pleomorphism is a particular theory about how different bacteria relate to each other. So it's the idea put forward by Hallier and other people that there exists a basic fungal entity, a so-called Pilzwesen in German, which can grow into different morphs. So it can grow into fungi, into yeast, and into molds under changing environmental conditions. So depending on the environment, this Pilzwesen, this entity, grows into different things. And each of these morphs causes fermentation. And that is, again, a very wide term that can mean alcoholic fermentation, can mean it produces sour milk, putrefaction, and also various kinds of diseases. And what you see in the corner there is a picture, not by Hallier, but by Thomas Henry Huxley, which kind of illustrates that everything is basically related to everything. So what is interesting about Hallier is that he was trying to develop experimental techniques for the investigation of that approach and for the testing of his ideas. And he developed two methods and two technologies, actually. And he said they both need to be combined to study pleomorphism. One is that we need to trace the development of one particular organism, so one particular Pilzwesen, through the different stages to kind of confirm this idea of development through the different morphs. And the other one is to examine the products of fermentation of the particular kinds of microorganisms to show what kind of things they produce. And here are the technologies that he developed. So on the left side, that is the apparatus for the study of development. So you can see that this is this glass sphere with a culture dish underneath, and that helps you to both keep the air clean, and then this glass thing, this glass dome, can be lifted so that you can use your microscope to investigate various stages of development. The other apparatus is to study the process of fermentation, where the point is that you have to make sure that nothing from the outside gets into your culture and contaminates it, because you want to establish what the product is of this one particular kind of organism that you have under observation. So he suggested these kinds of technologies to confirm his notion of pleomorphism, and unfortunately they didn't work, which from the perspective of today we can easily see why, because they did not what they were supposed to do. First of all, it was impossible to just keep one organism under observation and follow it through the different stages. And the other thing is that the apparatus just didn't keep contaminations out of the samples. So the thing is that better technologies could not be developed at the time. So people were, in fact, it's only recently that biologists have the technology to study development in the way that was envisioned in the 19th century. And it also didn't work to keep the cultures clean. So these two apparatuses did not do what they were supposed to do. And what is interesting is then there was another bacteriologist at the time who developed an interesting alternative, namely a physiological classification of bacteria. So he agreed with Hallier that bacteria are too small to be classified, which you can kind of see on the plate on the right side, because this is what you see with a microscope. You can't really distinguish these little dots. But Ferdinand Kohn and his co-workers suggested that bacteria can be classified by function. And so they distinguished three different types of bacteria. The first one producing colors, the second one producing fermentation, the third one producing disease. And what is interesting here is that they picked the first one to investigate because you could see quite easily with a microscope what kinds of colors these bacteria produce. So the idea is that regardless of the environment, you could see that the same types of bacteria always produce the same colored pigments, and on this basis you can actually produce a classification for different bacteria, but according to a very different principle. We are no longer classifying by form, we are classifying by physiological function. The other thing that I think is also important to notice is that this classification was not complete because, again, for the bacteria producing diseases, no real classification system could be found at the time. So this was a new approach to the same problem. Now, what exactly can we learn from these two examples, or rather from this one episode, for our conference theme? So the question is, was pleomorphism really a failure, and if so, in what way? And I think what we can see here, I should say that I'm, of course, generalizing over one example, but I do think that this is a fairly typical episode in the history of science. So what we can see here is that the debate at the time, when we look closely, turned out to be very complex and multifaceted. One thing that I think is interesting here, this is kind of in line with the typology that we saw before. I think when we look at these kinds of failures from this history of science perspective, it's very important that we distinguish between different kinds of failures. So failed theories, failed methodological approaches, failed implementations of approaches. And when we use that to look back at the episode, we see that the experimental tests of pleomorphism turned out to be impossible with the technology available at the time. The theory as such remained an option, not only for Hallier, who continued to believe in it, but also for many other people. I already showed you the plate by Huxley, which was several years after Hallier did his experiments. Hallier's observations inspired contemporaneous theories of wound infection because he saw bacteria everywhere, and that was actually something that other people took up to say, well, you know, they might play a role actually in some medical conditions. And then finally, the methodological principles, and I think that's really an important issue, the methodological principles of causal experimental research that Hallier took for granted were never contested. So it is the implementation of a certain technological approach that really failed here. So in fact, I think we can say that by these different approaches that I presented here very briefly, the methodological principles of causal experimentation were in fact confirmed. So let me see. When we look more closely at the historical situation, Hallier's scandalous and absurd failure doesn't seem to be such a spectacular failure after all. It's certainly not a retarding advancement or pure fantasy, unfortunately refuted, absurd, and so on. But the interesting thing is when you look at the way in which that judgment came about, you can see that at the time, one of the opponents of Hallier, who had a different theory, put forward this judgment, namely it was absurd, there was a pleomorphic extravagance, and it belongs to the scientific chronique scandaleuse. That was in 1887, 20 years after Hallier, and I think we can see that many people just kept referring to that without looking at the actual situation. So, what, if anything, did the 19th century bacteriologists learn from Hallier's failure? This is something that I think we will discuss. I think that they did not learn from the failure as such. Even though it was clear that Hallier's technology was not working, just by staring at the technology, you can't really find a way forward. So I would say that just as there is no royal road to discovery, there's also no secure path from failure to success. So my last kind of concluding slide, what I think we can do is we can learn from studies of scientific episodes like the ones I presented and like the ones that we've seen earlier. We'll usually find that the big failures, the so-called big failures, will turn out to be multi-layered, very complex events. We will also find something that I couldn't really show here in much detail. The day-to-day research practice involves numerous dead ends, detours, failed trials, partial successes, and delayed effects. And that kind of leads to my last and final message, namely that studies of actual scientific episodes can be extremely instructive, I think, if we pay close attention to the tools and practices of knowledge generation, not just the results, and if we consider why past commentators may have been inclined to be critical. And finally, trying to present a historical episode in less than 15 minutes defeats the purpose. Thank you.